Okay, welcome everybody. We're really excited that you're able to come out today. We are a group from the Yale Science Diplomats, which is a group of graduate and professional students, as well as a few postdocs at Yale University who are interested in sharing the science that we do with the public. And so we have a series called Science in the News, where we come to a series of different libraries and we give presentations on various different topics. And today, as you all may know, we're talking about big bacteria, big pharma, big problems. What you should know about antibiotics. So a quick disclaimer, we are not medical doctors. We obviously can't give you consultation in your own personal health. You should contact a medical professional about anything with that. We also have a dedicated time for questions and answers at the very end. So if you have questions during the presentation, we just ask that you hold them until the very end. But besides that, let's get started about going over what we'll be talking about in the talk today. So first, just a little bit more about us. Our first speaker, Emily Shine, is a third-year graduate student at Yale working in microbial pathogenesis. Her research focuses on small bacterial molecules and how they influence human health. A fun fact about her is she used to be a ballet dancer. Following that is going to be Melanie Martin. She's a postdoctoral scholar in the anthropology department at Yale University. Her research focuses on human biology, growth and development, predominantly in the infant through childhood region or age group. And she also focuses on nutrition and how these all affect the public health sector. A fun fact about her is that she is actually a former roller derby skater. And actually, unfortunately, our third speaker, Ramya Pathuri, is not able to attend with us today. She had a personal issue that she had to attend to in Boston this weekend, and so I will actually be giving her presentation today. But the script is entirely from her, and so I am just a conduit for her voice. I will not be able to answer questions on it, but perhaps Melanie and Emily will be able to do that at the end. But Ramya, who wrote the third part of our presentation today, is a first-year graduate student working on her PhD in molecular biophysics and biochemistry at Yale. Her work focuses on RNA regulatory elements, and she really enjoys cooking. But enough about us. What are we going to be talking about today? So first, Emily is going to guide us through exactly what antibiotics are and what superbugs are and how they emerge. After that, Melanie's going to be talking about how antibiotics affect us every day and what it means for our public health. And then finally, I'll be talking about what is in the future of drug development and what's being done for the future of antibiotics from the perspectives of governments, large pharmaceuticals, and academic research. But to get things kicked off, please help me welcome Emily Shine as our first speaker.
Hi, everyone. Thanks so much for coming out. It's a beautiful day. Thank God winter is over. All right, so today I'm going to lay down the building blocks of what antibiotics and superbugs and resistance is. So perhaps you've heard about superbugs in the news. There were some headlines around the approaching Rio de Janeiro Olympics where they found superbugs in the oceans surrounding Rio de Janeiro. It was a big public health issue leading up to those games. Perhaps you've also seen some instances in which individuals will have antibiotic-resistant infections that they can't treat, or they find new different types of antibiotic-resistance infections. And you might think, what generally does this mean for me, and how do superbugs happen? Where do they come from? So superbugs are bacteria that are resistant to multiple antibiotics. And so I think we're all pretty familiar with what antibiotics are. They are drugs that kill bacteria. And the way that we can visualize how antibiotics work in the lab is on a petri dish. So seen here is this disc that is filled with this nutritional jello, essentially, that we call agar. And this is the food that the bacteria will grow on. So when bacteria grow on a lab in a petri dish, they grow up as these little tiny colonies, these little isolated dots. So those white dots that you see there in the picture are the bacterial colonies. Now, if we have a different petri dish where we add in a drug, the antibiotic, along with the nutritional agar, no growth will happen because all the cells will die. So this is a simple way to visualize how antibiotics work. They kill bacteria and they treat bacteria. And I think because we take antibiotics so often, we tend to not appreciate how absolutely essential they are for our health system. Before we had antibiotics, about 5 out of every 1,000 women who gave birth died from an infection that they acquired during labor and delivery. About 1 out of 10 people who had any type of skin injury would actually lose a limb to that skin infection. And about 3 out of 10 people who contracted pneumonia died. And a host of other relatively simple diseases that we can treat today were virtually untreatable. So it's essential to remember how important these drugs are. And we can owe this revolution to healthcare to one man named Alexander Fleming. And he was a Scotsman, and he is responsible also for one of my favorite stories in microbiology and in science in general, because he illustrates the virtues of laziness in science. So what happened was Alexander Fleming went on vacation or on holiday, and he left out his petri dishes out on his benchtop. And so they were uncovered and they were able to be contaminated by the air. So what happened is when he came back, he noticed that there was this fungal spore forming on his petri dish that was supposed to have only bacteria on it. So that white spore there at the top is the actual plate, the actual fungal spore on Alexander Fleming's plate that he took a picture of. And he noticed very astutely, before just throwing it into the trash, that around that fungal spore, there's really no bacterial growth. If you see anything, those bacterial colonies that I mentioned are very, very small. So he hypothesized that this mold was making some sort of substance that was inhibiting the bacterial growth. And he turned out to be right. And so later, scientists were able to isolate that compound that the mold was making, and that's actually penicillin, what you use today to treat wisdom teeth infections or any type of infection. So after we discovered penicillin, we didn't stop there. This is a timeline showing you of when all the different types of antibiotics that we found after the discovery of penicillin. And these include things like erythromycin and ciprofloxacin, which are both used in the clinic to treat respiratory infections. And actually, if you ever get a prescription from your doctor, and if you notice that on the box has a drug name ending in mycin or in, that's a very, very common suffix for an antibiotic. So that clues you in onto the type of drug that you'll be taking. So you might ask, why did we need so many different types of antibiotics, right? Well, there's obviously multiple ways to skin a cat, and there's multiple different ways to kill bacteria. So in the case of penicillin, for instance, penicillin's target is the cell wall. So the cell wall of the bacteria is what it sounds like. It's a cell wall that protects the bacteria, keeps all of its containment inside, and it protects it from any outside invaders or any environmental stresses. And so when you inhibit the synthesis and you can't make that cell wall, it breaks down that barrier and the cell will die. Erythromycin, on the other hand, which is another example of an antibiotic that I gave you back on the last slide, works in an entirely different mechanism. Erythromycin targets the protein factories of the cell. And so when those protein factories are essentially shut down, the bacteria can no longer make its goods that it needs to survive. And so that is a way that the cell can die. The last drug that I mentioned on the last slide, ciprofloxacin, again works in an entirely different mechanism. And that will target the cell's instructions for how to live. And those instructions are essentially the cell's DNA that we'll get back to later in the talk. So what happens is when you target those instructions, and more importantly, when you can't copy those instructions to give to your neighbor or your daughter cell when you grow and divide, those cells will not be able to survive. So in this instance, this green bacteria here denotes that this bacteria is susceptible to killing by all 3 of these different types of antibiotics shown here. Now, as you can imagine, not all antibiotics are going to be able to kill all different types of bacteria. So for instance, there are some bacteria out there that are able to make molecular scissors, which target penicillin. And penicillin, as I mentioned before, is the drug that targets the cell wall synthesis. So when certain bugs are able to make their molecular scissors, the penicillin actually gets cut up and it breaks down and it can no longer inhibit cell wall synthesis. Showing here. So in that case, the cell walls remained intact. And so this bacteria doesn't care that penicillin is in the environment. And that's what we call resistance. This cell is now resistant to killing by penicillin because it's able to destroy that drug. Now, molecular scissors don't happen to be available for ciprofloxacin. So in this case, when ciprofloxacin is in the environment, there's no way for the cell to overcome that pressure. So the cell's instructions will be targeted, you can't copy those anymore, and this cell will die. So this generally demonstrates what resistance is. So I'd like to really take this opportunity to describe what resistance isn't. So a lot of people get confused when we talk about antibiotic resistance, and you think that your own body is becoming resistant to the drugs, and that's not true. Antibiotics only work on bacteria. They don't work on your tissues. So it's the bacterial infection that you have that's actually becoming resistant to your drugs. So again, you're not becoming resistant, it's the bacteria inside of you that are. So to model how resistance spreads in an infection, let's say we have a population here hanging out in someone's bloodstream of green bacteria that are susceptible and red that will be the resistant ones. So when we apply penicillin, our green ones, which are susceptible, will die off as predicted. The problem, though, is that the red one doesn't care that penicillin is in the environment because it has those scissors. So the red one is free to conquer and divide, essentially. And so now you get a brand new infection that your last drug doesn't treat at all. So normally what happens is doctors just use another drug, ciprofloxacin in this case. So in this case, luckily we can clear this infection. But as you can imagine, there are multiple different types of resistance mechanisms too, and just using another drug is not always going to work. So for instance, this blue bacteria here. Let's say that this one is resistant to penicillin because it has the pair of molecular scissors that cuts it up. And so our cell wall will be intact. But let's say this also makes a pump. So some bacteria can make a pump, which means that anytime a drug can get in, it immediately gets flushed back out. And so the ciprofloxacin can't target the cell's instructions anymore. In this case, you have a bug that's resistant to both of these drugs, which is a major problem. And we call this a superbug. And Superman is on there to denote special powers, but this is not a good thing for humanity. This is a very bad thing. So how do bacteria become superbugs? This can happen usually in one of two ways, one of which is very, very quickly. So let's say, again, we have an infection in a bloodstream, and this red one here is resistant to penicillin. It doesn't care that penicillin is an environment. But let's say next to it is a purple bacteria that's resistant to ciprofloxacin. What can happen sometimes is that these bacteria are in close contact with each other. They can essentially share the information of how to build a pump or how to make a pair of molecular scissors. And so overnight, these can become superbugs because they transfer this information and they become resistant to both of these drugs. Other times, this process is not so fast and it's a lot more gradual. And it's kind of akin to weight training. So when you go to the gym, you don't immediately start with the heaviest weights. You start from the bottom and you train upwards. And that's essentially what bacteria can do. Bacteria can get used to small amounts of antibiotic in increasing amounts, and they can slowly tolerate larger and larger and larger doses of antibiotic. And to illustrate this, I'll give you an example of this here, which is a green bacteria, but it has a pump. But in this case, even though it has a pump, this bacteria is still susceptible to killing by this drug. Because what happens is this pump is actually pretty bad in this case. So when the drug gets in, it spins around, it takes a long time to get flushed back out. So the way that you can overcome this is by applying a very, very large dose of the drug. So when you have a large concentration of a drug in the environment, it overwhelms the pumps and it can get in and kill the cell. So this happens very naturally, natural tolerance of a drug. And this is why it's incredibly essential when you get an antibiotic prescription from your doctor that you take the full dosage at the highest dosage all throughout, even if you think that you're starting to feel better, because you want the highest dose to kill any of these guys that can somehow tolerate tiny amounts of drug. So by weight training, what happens with weight training in getting used to larger and larger doses of antibiotic is actually through changes to the cell's instructions. And I talked about this before. These cell's instructions are actually molecules of DNA. And these molecules of DNA are made up of these chemicals that scientists call A, C's, T's, and G's. And these are letters, essentially, that are made up into words that the bacteria can read, and that gives them the instructions for how to live, how to make everything that they need, how to respond to environmental pressures, etc. So what happens is as they grow and divide, sometimes the wrong letters can be introduced into that sequence, and you'll get an entirely different type of word. Sometimes this is good, and sometimes this is bad. So the good mutations here essentially act by improving the pumps then. So the word that tells the cell how to make this pump can change over time, and so we have kind of a bad pump into a new shiny red Corvette, new and improved pump. So the gray pump, again, is the one that is very slow and takes a long time to flush out that drug, whereas this guy immediately flushes out its drug. And so this becomes a game, then, of as bacteria are growing and dividing, of may the best mutant win. So we have our susceptible bacteria with the bad pump, and as it grows and divides, you can see we get different colors of pumps, and that denotes how efficient they are at pumping out the drug. And so in this case, if you have a small amount of ciprofloxacin, that doesn't really affect anything, but as you increase the dosage of ciprofloxacin, those will die. The ones that are not as tolerable of this drug will die off. And when you get to large and large concentrations of this drug, you leave the red one behind that has the best capability of flushing out that drug. And so that one is able to grow and divide and conquer. And this is evolution in action. We can see a selection for the best bacteria, or in this case, best meaning the best one that can pump out that drug. And that is the one that wins in this competition and is able to keep continuing onward. And we can see this evolution in action in the lab very easily because bacteria takes very, very little time to divide. So the Petri dish idea that I mentioned at the beginning, some scientists at Harvard did a really cool experiment where we built a Petri dish that is 2 feet by 4 feet. And you can imagine this as set up as a football lab, a football field. So what they did is they had no antibiotics at the end zones, and increasing towards midfield, they had increasing amounts of antibiotic. So you go from no antibiotic to 1,000 times the antibiotic right in the center of the field. And they plated the bacteria starting at the end zone, and you can see as the bacteria are growing and dividing, and some of them have mutations introduced into that DNA or their cell's instructions, you can see new mutants arising that are able to tolerate larger and larger doses of antibiotic. So I have a short video. So this is the setup field again. So again, at the end zones is 0 amounts of antibiotic and 10 times more antibiotic as you increase to midfield. And so this Petri dish is black in this case because it has ink, and the bacteria will appear as white in order for you to visualize it. So the white is the bacteria that is plated then, and it stalls as soon as it meets the threshold of where there's antibiotic present because it doesn't know how to overcome that. Now that arrow there is a mutant that has arose that is able to get a better pump, for instance. And that's able to grow and spread, and you get a ton of other mutations as well. And again, this is happening because bacteria divide so quickly that mutations can be introduced into the bacteria very, very quickly. And again, you can see a stalling when they hit 100 times more antibiotic, but again, they're able to overcome that through successive mutations. Until finally, they reach the threshold in which they can tolerate 1,000 times more antibiotic than they did before. And what's really incredible is that this entire time lapse that you see takes place over 11 days. So in 11 days, which is essentially the length of an infection or how long you're taking antibiotics for, you can see bacteria go from completely susceptible to antibiotic resistance. So it's pretty cool for us scientists to go and see that, but it's pretty scary for the rest of us. In fact, there was earlier this year, there was a story in which a Nevada woman had an infection that she died from, and this infection was resistant to every single antibiotic available out in the clinic, all 26 of them. And we're getting to this point where we're seeing these superbugs that are resistant to multiple antibiotics more and more and more. And how do you treat an antibiotic or how do you treat a bacteria that no antibiotic can kill? So Melanie is going to talk about how we got here and how some of our own actions have contributed to antibiotic resistance spread.
Okay, so I'll be talking about, as Emily said, sorry, there we go. Now that you understand a little bit more of how antibiotic resistance evolves, what this means for our health and our public health system. I'll also be focusing on aspects of our society and lifestyles that have contributed to the current public health crisis, and then for that reason, some things that we can do to potentially overcome this crisis. So I'll start off by emphasizing that this is indeed a health crisis. Sorry. In 2014, the CDC named antibiotic-resistant infections one of the top 5 health threats in the U.S. Each year, about 2 million people in the U.S. alone get sick with infections caused by antibiotic-resistant bacteria, and 23,000 of those people die from those infections. Now, one of the most common culprits in this is a superbug that you might have heard of called methicillin-resistant Staphylococcus aureus, or MRSA. Now, Staphylococcus aureus is normally what we call commensal bacteria. So it's a bacteria that inhabits our body normally but doesn't cause us any harm. It inhabits the nose and upper respiratory tract. Now, what happens sometimes, though, is that you might get a cut or a wound or some other sort of skin lesion, and that Staphylococcus aureus can travel from where it normally resides to another part of your body where it's not supposed to be. When that happens, that's called a staph infection, and they can be very nasty infections. Now, MRSA is a breed or a specific strain of Staphylococcus aureus that has become resistant to many different types of antibiotics. So what happens is if you are a carrier of MRSA, and many people are, then if you get a cut or an abrasion and the MRSA travels from where it normally resides into where it's not supposed to be, now not only do you have a really nasty staph infection, you have a staph infection with a bacteria that can't be killed by most antibiotics out there. And this can be very dangerous and often lethal. So this creates many problems for our health and our healthcare system, not only because of the individual antibiotic-resistant infections themselves, but because this means that we need new drugs to combat many infections. And as Jeremy will explain after I finish, that this takes a lot of time and money. There's also a whole side of our medical care system that depends on antibiotics. So for example, if you're undergoing surgery, you will probably be given antibiotics as a prophylactic measure, so to prevent infection while your body is so vulnerable. Same thing for many people undergoing treatment for chronic diseases such as cancer. Their immune systems are suppressed, so they need to take antibiotics continually to prevent a bacterial infection. So as more of our common antibiotics become ineffective because they are evolving resistance, this means that many of the modern medical marvels that save so many people's lives are also at risk of becoming ineffective. Now, interestingly, there's another side of just taking antibiotics that I think that many people don't appreciate, the negative effects of just taking antibiotics. For example, there's actually many side effects to taking antibiotics, even when you've been given them for an infection or preventative measure. So diarrhea being one. They can also interfere with the efficacy of other drugs, that is, how well other drugs do their job. And antibiotics can cause allergic reactions, from mild to severe allergic reactions. In fact, 1 out of 5 emergency visits are due to adverse effects of antibiotics. Another way that antibiotics can have harmful effects on our health every day is through their effects on the gut microbiome. So we all harbor trillions of bacteria in our intestines. This is collectively called the gut microbiome. And these bacteria are commensal, meaning they just reside there, they don't do any harm, or many of them, in fact, are beneficial. They provide a lot of benefits for us, their hosts. And this is, in fact, a reason that so many yogurt manufacturers promote eating yogurt for your gut health, because yogurt is chock full of these beneficial bacteria like lactobacillus and bifidobacteria that we want to populate our gut and provide us with many benefits. So one of those benefits is digestion, particularly of plant cell walls. So we can't break down the material of many plant cell walls, but the bacteria do. And then from that, they give us energy, they give us nutrients and vitamins. These beneficial bacteria also protect us against pathogens. So these are bacteria and other microorganisms that can cause disease. And they do this in 2 ways. First off is through a barrier protection. So For example, if a pathogen enters your intestine, it encounters all the beneficial bacteria that are inhabiting the walls of your intestine, so it can't really get a foothold. It can't establish, and therefore it can't replicate, and it's going to die off. At the same time, the beneficial bacteria also communicate to our immune system and help coordinate immune responses against those pathogens. So what happens then is that the pathogens will die off. Now, sometimes, though, a few pathogens manage to get through that barrier. They manage to get past our immune systems, and they'll be able to establish in the cell wall and replicate. And when that happens, then you do want to take an antibiotic for that bacterial infection. Now, the problem is, though, as Emily explained, antibiotics can target multiple bacteria that all share these same weaknesses. So this means that not only is that antibiotic going to kill off the pathogen, it's going to kill off many other bacteria that have the same weaknesses as that pathogen. Now, over time, after you complete the course of antibiotics, some of those beneficial bacteria they'll start to replenish. The problem is then, though, that they may not come back in the same numbers, and some species might be lost forever. And so this leads to a more depleted gut microbiome that is lower in bacterial abundance overall and lower in bacterial diversity. And increasingly, we recognize that low bacterial diversity in the gut is associated with a host of chronic diseases, including inflammatory bowel disease, autoimmune disorders such as rheumatoid arthritis, and even obesity. So it's interesting then that given the just the actual health risks of taking antibiotics, one of the reasons that there is this current public health crisis of evolving antibiotic resistance is because of overuse and misuse of antibiotics. And one of the main reasons for that is overprescribing antibiotics. So right now in the US, about 4 out of 5 Americans every year are given a prescription for antibiotics, but 1 in 3 of those prescriptions are completely unnecessary. And this is most often the case because those antibiotics are given for viral infections, so infections caused by viruses. These are many respiratory illnesses. Viruses are microorganisms that are not bacteria. They don't have any of the same weaknesses as bacteria, so antibiotics are completely useless against them. Now, I want to emphasize that there are, of course, many common infections for which antibiotics, they're caused by bacteria, so antibiotics are still very useful. And these would include strep throat, whooping cough, and urinary tract infection. And a doctor, if you go complaining of these symptoms, can safely prescribe you antibiotics for those infections. There's a whole other class, though, of illnesses such as sinus infections, middle ear infections, and bronchitis and chest colds that can actually be caused by bacteria or viruses. But doctors will often just prescribe antibiotics to be safe or because of the cost of performing the test to see whether the infection is actually bacterial or viral to begin with. Then there's a whole class of our most common respiratory illnesses, so the common cold, sore throat, and flu, which we know are caused by viruses, not bacteria. But in many cases, doctors will still prescribe antibiotics to patients that present with these symptoms, often because the patients demand them, and the doctors just want to please their patients. Now, a second reason that's contributing to the misuse and overuse of antibiotics and therefore this public health crisis is actually agricultural use. So right now, about 70% of all the antibiotics produced in the US are actually not given to humans, they're given to livestock. So these are the animals that we raise for our own consumption. And they can be given because outbreaks of bacterial infections can happen and you want to get those animals better. But it's also important to point out, though, that outbreaks are very common in our modern industrial farming practices, often because of very crowded and unsanitary conditions. And in fact, many farmers give animals antibiotics prophylactically, that is, to prevent infection, by giving them low doses of antibiotics continuously. Now, the other reason that antibiotics, though, are given often in farming is because since about the 1950s, farmers have recognized that giving animals antibiotics makes them grow faster and get fatter. And that, of course, is good for their bottom dollar. So how does antibiotic resistance develop in the animals? Well, it evolves through all the same mechanisms that Emily described for how they evolve in humans. So this means that potentially on every farm, there's thousands of animals that can be walking around with antibiotic-resistant bacteria in their intestines. Those get shed out through feces, and then they can spread to other fruits and vegetables that are growing on the farm. They can be in the meat themselves, so from other animals that we're going to eat. And then when we buy and prepare and consume these foods in our old households, they spread to us. So, understanding how our lifestyles and society have contributed to the misuse and overuse of antibiotics, we can all actually individually take some steps to help combat this. The first would be don't take antibiotics unnecessarily. If you have a cold or a flu, don't go to your doctor demanding antibiotics. The other thing that you can do is support restaurants and support, whenever you can, buy animal products that have been raised from animals that have been raised without antibiotics for growth. Increasingly, there are a number of restaurants, even popular restaurants, that have committed to this type of reform. In 2016, the National Defense Consumer Resource Council, along with some other consumer agencies, put together this sort of report card showing how well different restaurants were doing in terms of their commitment to reform. That means they promised to only source animals or animal products from farms that were not giving antibiotics to animals for growth, promoting purposes. So doing the best here would be Panera, Chipotle, Subway, and Chick-fil-A. Middle of the pack, McDonald's and Taco Bell. And then, unfortunately, there's still a whole lot of other restaurants that have not made any inroads to that. Although I should say, I actually heard on the radio yesterday that KFC had just committed to using antibiotic-free chickens and phasing. By 2018, they would completely phase out the use of chickens that were raised with antibiotics for growth. Okay, so just to recap then, please remember that antibiotic resistance is a public health crisis. It threatens all of us. We have contributed to this mess through individual lifestyle and societal factors, but for that very reason, we can be part of the solution. And so now Jeremy is going to explain how government, industry, and academic research are working on other ways to come up with solutions for this problem. Thank you.
Thanks, Melanie. So far, we have heard about how antibiotics work and how superbugs develop, and also how they affect our daily lives. I wanted to focus in on sort of the new hope for antibiotics in the future and how pharma, our government, and academic researchers are all combating the problem of antibiotic resistance. So I wanted to start so let's start by talking about what big pharma companies have been up to in this whole story regarding antibiotics. So this is actually the same timeline that Emily showed earlier in the talk today, but I wanted to show some other features of this graph. So first, you can see that most of the antibiotics were discovered in the period from like the 40s to the 60s. And at this point, pharma companies were able to very successfully profit from many of these antibiotics, such as penicillin. After that, though, there's a large gap that there weren't a lot of antibiotics developed, and then in the 2000s, a few more were developed, but the number that were actually discovered was still pretty low. The bottom of the timeline shows actually when the first bacteria were discovered that were resistant to these corresponding antibiotics. And so just to kind of cut to the punchline, every antibiotic that's up here, we've found in some sort of bacteria that is in fact resistant to it. But not only have we found resistant bacteria to all of these existing antibiotics, but the ones we've discovered more recently, over here, we've found resistant antibacteria even faster than normal. You can see that the brown one over here, daptomycin, we were able to find bacteria that were resistant to it within just basically a year. And so just as a recap, each of these antibiotics has some sort of resistant bacteria to it, and we're also finding these resistant bacteria faster than ever before. So given this problem, what is pharma doing about this? Well, big pharma companies aren't really investing in antibiotics the same way that they used to, and they aren't for a few reasons. The first one is that antibiotic drug discovery is a really hard thing to do. Whenever they're submitted to the FDA, for example, only about 1% of proposed antibiotics that are sent to the FDA are found to actually be viable, safe, effective. Only about 1% of them are able to make it through this pipeline. This is in comparison to other types of drugs, such as like diabetes medication or blood pressure medication, that 7% of them are, roughly 7% of them are able to make it through. This is just an example to show how difficult it is to actually develop antibiotics, because antibiotic drug discovery is really hard. Another reason for the lack of investment in antibiotics is due to the really high cost of making antibiotics. So just in general, the development of a single drug usually costs somewhere between $400 and $800 million. And so for antibiotics, a typical dose only lasts from about 7 to 14 days, so 1 to 2 weeks. This is in comparison to other types of drugs like diabetes medication, for example, that patients have to take for the rest of their life. And so just by looking at this, you can see that there's just a lot more money to be made from developing these longer-term drugs, because these are drugs that people are going to be using for basically the rest of their life, as opposed to these ones which are only used for 1 to 2 weeks and really only as a last resort. So that's one reason why there's been less investment in antibiotics from a pharmaceutical perspective. So how do we drive new antibiotics research? Well, without investment from pharmaceutical companies, how can we continue to find these potent antibiotics? There are a few mechanisms to do so. One of them was implemented by the Obama administration in 2012, called the GAIN Act. There's another, is ongoing innovative academic research. And so I'm going to talk about both of these, but first I wanted to talk about the GAIN Act and what that is. So the GAIN Act, or the Generating Antibiotics Initiatives Now Act, was implemented in 2012, and it does two main things to spur antibiotics in development. The first is that it allows the company that first brings the drug to market to exclusively produce it for 5 more years after it gets to market. So that was a mouthful right there that I just said. So let's kind of break that down. Let's break that down slowly. And let's use Tylenol as an example. So let's say that a pharma company discovered the main ingredient in Tylenol, and at that point, that's when they file a patent which lasts for 20 years, and they file it whenever they discover it immediately. So that's in place so that it protects their invention. That's when the clock starts ticking. After filing a patent, the company can start development of the developing the drug, meaning clinical trials, formulation testing, and finally going through the FDA approval process. The drug development and the FDA approval process altogether can take anywhere from 12 to 20 years, and by the time the company finally has a marketable product, like Tylenol itself, the protected period in which the company could make money would basically end. What the GAIN Act does is extends this amount of time. So the GAIN Act basically extends this amount of time of exclusivity by 5 years to try and motivate more drug companies to investigate antibiotics and be able to actually get some sort of profit from them. So without the GAIN Act, if it took them 20 years to produce it, they've basically done all of the hard work, and then anyone else can come in and basically make the generic brand of Tylenol and make all the same money without having to put in the money to invest it. And so one of the things that the GAIN Act does was allow basically an extra 5 years for the companies that produce the drugs to have exclusive rights to it. And so that's one way to help incentivize the production of antibiotics. So they can make more money. The other benefit granted by the GAIN Act is the ability for new antibiotics to be on the FDA fast track. So in the Tylenol example that we just talked about, the actual FDA approval process takes somewhere around 2 years to go through. However, when there is an unmet need in the nation, say the rapid depletion of potent antibiotics, the FDA designates the drug to the fast track to allow more frequent meetings with the FDA and also faster communication. Basically, the drug gets greater access and priority at the FDA, meaning faster approval. So I want to emphasize that this is not saying that they're like cutting the clinical trials. It's not that they have less stringent requirements. It's just that they dedicate more people to looking into this, and so they're able to get it through this FDA approval pipeline faster. It's not less, it's just going through at a quicker rate. And so because pharmaceutical companies are able to get greater access and priority at the FDA, it leads to faster approval, which again leads to more money for the pharmaceutical companies themselves. And just to show some examples of some companies that have actually used the GAIN Act to produce antibiotics, these are a whole list of different antibiotics that were created because of this GAIN Act. And so even if you don't recognize all of these antibiotics themselves, I'm sure that you might recognize at least some of the companies such as Merck or AstraZeneca right here. So these are real drugs that were produced fairly recently because of the GAIN Act. So the GAIN Act and basically what the government was able to do is create some of these drugs. But let's talk about another way that we can help drive the development of antibiotic research, and that is through academic research. And so a really nice example of this academic research in action comes from Dr. Kim Lewis's microbiology lab at Northeastern University. The lab here studies antibiotic resistance in bacteria, and through this research actually identified an antibiotic called tioxobactin, which was discovered in 2015. Knowing how quickly we have been discovering bacterial resistance and how quickly bacteria can evolve resistance, it's remarkable that we haven't found any resistance to tioxobactin yet. And so as I said, most of these other drugs, most of these other antibiotics were able to find these antibiotic-resistant drugs very quickly, but we haven't found anything for tioxobactin yet, which is a really exciting jump forward in the field. And so let's actually walk through some of the details in this really amazing story by looking at the academic paper that discusses this. And we'll talk about how the Lewis Lab actually made this discovery. So the story starts in the soil. Unseen to the naked eye, there are actually millions of different types of microbes that live in the soil. These microbes help each other and hurt each other in their fight to survive in the diverse soil environment. Most antibiotics have been mentioned so far have actually been discovered by examining chemicals made by soil microbes. Now before, when we didn't have many antibiotics, we could just leave a petri dish out and maybe find some bacteria. However, by and large, a key problem with examining soil bacteria is that it's really hard to grow them. If we can't grow them in isolation in the lab, we couldn't study them effectively. And so the tools that exist right now are like having a fishing rod that can only grab a few random types of fish. In fact, the tool I'm talking about is actually growing them on an agar plate in a petri dish. And using this, scientists are only able to really find and grow about 1% of the diversity of bacteria in the soil. That means that out of all the different species of bacteria that do live in the soil, we're only able to find about 1 in 100 of them using this method. So what we need is a better way to fish. And the Lewis Lab did just that. And so what they developed was basically a better way to fish for these bacteria. And what they developed, instead of using this fishing rod to get a single type of bacteria at a time, they have this net that they're able to use to get a lot of different types of bacteria. And using this, this net, which they invented called the iChip, nothing to do with Apple products, could capture 50% of the diversity of the bacteria in the soil. And so let's talk about how the device actually works. So the device works so well because it actually allows for the growth of individual bacteria that are isolated in these little cells. So you can kind of trap these individual bacteria in there. You can isolate them in these chambers. But it also allows for the isolated bacteria to grow in the soil and exchange nutrients with other bacteria that are in the soil. And so remember that the bacteria are dependent on these symbiotic relationships, and they depend on each other for survival. And so the iChip allows for the bacteria to keep doing what they do in the comfort of their own homes. This last picture here is really just a picture of the iChip in action. You can see someone's holding it just to get a scale of how big this iChip actually is. And so what did they actually find with these studies? And so from the iChip, many interesting bacteria were found, and one of them in particular is this one called Eleftheria terrae, which is a new bacterium that we've never been able to grow before using this Petri dish method. What they found was that this bacteria is able to actually make a compound called tioxobactin that can kill bacteria really, really effectively. So let's talk through two different experiments that show how effective tioxobactin can be. So in one experiment, a bacteria called S. aureus, which we talked about earlier in this talk, which is very similar to MRSA, was grown in these 4 different tubes here. The cloudiness of each tube means that there are bacteria growing in it. You can almost sort of see them in a sense. Each tube had a different antibiotic added to it. With these here. The one on the right here is the tioxobactin itself. But all these antibiotics are different antibiotics used commonly in the clinic today. The cool thing about this experiment is that after just a few hours, only the bacteria treated by tioxobactin, the one on the far right over here, resulted in cell death. And so, just to reiterate, only the tioxobactin was able to kill the S. aureus cells in these concentrations. In another experiment, the same bacteria, S. aureus, which we just mentioned, was treated with low levels of either afloxacin, which is one type of antibiotic, or tioxobactin for several days to try and see if the bacteria develop resistance. So as Emily mentioned before, you can kind of train these bacteria to become resistant with larger doses of antibiotics. And so if you look over here, this is basically the amount of antibiotic that was needed to kill the bacteria that were in there. You can see that this afloxacin, as time goes on, you need more and more of this antibiotic in order to kill them. But meanwhile, the tioxobactin over here, you can use the same low concentration the whole entire time, and it didn't seem like the bacteria were getting more and more resistant to it. And so for this particular one, it was almost as if the bacteria weren't getting any stronger with its resistance. So these two experiments, this one and the one I mentioned on the last slide, both use S. aureus, but low levels of tioxobactin have also been shown to effectively kill MRSA, and also the same bacteria that produces anthrax, which is really great that that can be done. And so let's talk about exactly how tioxobactin works. So it's interesting to think about why this is so useful and why many scientists think it's better than a lot of other options we have out there. So here's the slide that Emily presented from before about how antibiotics work. So tioxobactin acts on a key building block of the cell wall, a molecule called lipid 2. But unlike penicillin, which also works on the cell wall, it inhibits a molecule made very early in the process rather than later. And so it's kind of like pulling out the bottom piece of a Jenga tower, as opposed to penicillin, which is pulling out ones from a random place in it, or maybe even the top. And so because the tioxobactin is able to get a much more fundamental part of the cell wall, this is why scientists think that it's going to be extremely difficult for bacteria to evolve and develop a resistance to tioxobactin, because it affects it so early in the process. It just doesn't have time to get resistant. So what exactly is the future of tioxobactin? Well, Teixobactin treatments are currently being developed by a company called Novobiotic, which is a company that was founded by Dr. Kim Lewis, the person who discovered this in the first place. And it'll actually be going through the drug development pipeline at the FDA relatively soon, which is really exciting. So, just in conclusion, so while we spent a lot of time today talking about how antibiotic resistance is a growing problem in our world, I'd like to end by emphasizing how important antibiotics really are and what they've been in the progress of humankind. So penicillin, the very first antibiotic discovered, really changed the world. And since then, we've managed to make many bacterial infections almost harmless. And we owe our modern civilization to antibiotics and the important work from Alexander Fleming with this book called Penicillin Man over here. It's important that we use antibiotics wisely and continue to research new ones to sustain our future. And so I want to thank everyone for coming out today and learning more about antibiotics. I hope that between Emily, Melanie, and myself, you've learned about how antibiotics work and what superbugs really are, how their increased resistance what their increased resistance means for our public health, and also what types of progress are being done to stay ahead of antibiotic-resistant bacteria. So with that, I'd like to say thanks to everyone for coming. I'm going to bring our two actual speakers up today to answer any questions that you may have. So thank you for having us.
You mentioned this, the dynamic of how the bacteria plays with, pushes up against its environment in order to increase its resistance to various factors in the environment. But one thing that doesn't seem to be explained by that way of looking at things is the damage that bacteria does to the host animals that it's on. I mean, there's bacteria in the dirt, and you're assuming it's not doing so much damage to the dirt, but if you kill the host animal that you're on, you basically don't get to propagate. Is there any study of that sort of as an inhibition mechanism? How does that
All the time, yeah. So that's a huge field in microbiology. In fact, a lot of really difficult infections, like tuberculosis, they make their living just by evading host infection because they know that the host spots them and you clear it out, that's not good for them. They also don't want to kill their host. So tuberculosis is actually a very difficult infection to treat because it suppresses your immune system and it evades all of that. So that's definitely a strategy that bugs use all the time in order to keep living. Salmonella, for instance, if you get food poisoning, salmonella is what we describe as a self-limiting infection, meaning that normally it doesn't kill you because that would be very bad for the salmonella. So it actually knows how to tune down its virulence or how bad it is technically as you progress through the infection. Did that answer your question?
Is that a strategy for learning how to defeat the bacteria overall? Because the self-attenuation, you can see if you could make that
not happen.
Yeah, like an increasing mechanism, it would attenuate itself out of existence.
I think what you're also getting into is using kind of immunotherapy, like using the host defense system to target your bacteria better. And that certainly is a strategy that lots of people have tried. A lot of research is in using gold nanoparticles, actually, which adhere to bacterial cells, and that helps the immune system know, hey, like, I need to clear this faster. It's just tricky because immune systems are very difficult. When you mess around with them, you might go all the way to the end of an autoimmune disorder, and they sort of have to things they shouldn't. So, and immunology is a very difficult field, and that strategy is difficult to kind of get the specifics out, and that's why it's a lot easier to use a small molecule drug like an antibiotic, but certainly workable.
I'll also just add to that, I think another aspect that you're touching on is really what, say, evolutionary biologists would study in terms of just evolutionary modeling of how hosts and different microorganisms co-evolve together, because this is something that's always kind of in flux. When you have a microorganism move to a new host, they can start out very virulent and something like that can happen. If they kill off too many potential hosts, they would have nowhere to go. So over time, less virulent species of that particular microorganism would be evolving where they kind of strike a balance, their virulence strikes a balance where their virulence factor is allowing them to spread and propagate but not kill off all the hosts at the same time. So, but that is a dynamic that takes time and isn't necessarily something that you can manipulate at the population level, and it becomes a threat anytime with a lot of, say, zoonotic diseases. Usually when there's a one, say, bacteria or virus that's usually associated with some animal crosses over to a new species, and this is what happened with Ebola, potentially, and then we haven't, in humans, there has been no ongoing evolution for lower virulence, and so it rapidly spreads through and wipes out. And nobody's willing to sit back and let the evolutionary dynamics of that play out and see over time would Ebola evolve lower virulence.
My understanding was that one of the ways we get resistant to antibiotics is by eating chicken or beef that was treated with antibiotics. Because it's just like taking antibiotics in small amount. The meat itself was contaminated. I didn't get that idea from what you were presenting. Was I wrong for thinking that the meat itself is contaminated?
So the meat itself can have, if you don't, say, cook it all the way, and there's still live bacteria in it, and those bacteria are resistant, then you can get that infection.
No, I thought it has antibiotics in it.
Like in the muscle tissue?
Right. And that is affects our cells by, it's just like taking a small amount of antibiotics ourselves.
I'm fairly sure FDA standards do not allow antibiotics to be present in the meat itself. So they should go through screening to see if there's antibiotics in there. Whether or not the industry standards are adhering to the FDA rule, I'm not sure about. But I'm pretty sure it is illegal to have large amounts of antibiotics.
It's clear, the chicken itself is clear?
Yeah, it should be, according to the FDA.
Right, like when you take antibiotics, you shouldn't have trace antibiotics in your muscle tissue.
But also, like, I mean, it would be bad if any pharmacological drug was present in meat, because who knows how you might get side effects from that meat then. Like, that's a very bad thing. Yeah.
So you talked about the microbiome. Is there a scientific consensus as to what the optimal microbiome environment is for us? And
Not at all.
Along those lines, are there tests that you can, let's say you take a bunch of antibiotics and it has a negative effect on your natural microbiome. Are there tests you can do to see what you're lacking, what you should be replacing? And one final part of that question, are any of these new, more effective drugs being developed to just target bacteria that are not supposed to be there?
Well, in answer to the microbiome, the only consensus really at this point is that when we look at different populations of humans, when we look at populations, say, of animals in captivity versus the wild, you see differences in that the less kind of sterile the environment, the less typical Western diet that people are given, or even mice are given, those hosts have more diverse microbiomes. And at the same time, we also see lower rates of chronic disease. So from that, the consensus is a more diverse microbiome is healthier. And this is all associative, not, are we able to show the exact causal mechanisms leading one to the other? And there's definitely not a lot of consensus in terms of what particular species that diversity should be made up of. Different studies will show different associations of some bacteria and some disease, or some bacteria and kind of healthier outcomes, but it's definitely far from certain. The one thing that I could say in terms of there's probably companies, I think there definitely are companies out there right now that you could send your own stool sample to, and they will give you back a read on the diversity and different levels of abundance of different bacterial taxa and species. Now, what you're really going to do with that, I don't know.
Actually, though, to add on to that, the idea of a poop bank is actually not that crazy of an idea. Like, for instance, if you know you're going into surgery and you'll be taking antibiotics or whatever, it might be a good idea for you to save a stool sample, because whatever your microbiome is before you introduce an antibiotics, doctors can use something called a fecal transplant, where they actually put in a piece of your poop back into your intestine, and that can repopulate whatever you had before back in.
It's actually a very promising therapy for a lot of people who do suffer from inflammatory bowel disorders and Crohn's disease too, is to do fecal transplants. And I will say that there's probiotics too. I mentioned yogurt. There's also the probiotic, you can take over-the-counter probiotics. And there's not very good evidence that those work so well. I think except for following a course of antibiotics or like gastrointestinal infection where you do end up with a depleted microbiome, those would help you rebuild a healthy, more diverse
I think
gut.
actually the conventional wisdom too is that if you have a disturbance in your microbiome, like if you're taking antibiotics or if you have like food poisoning or something, to continue, like once you've gotten over that illness or once you've stopped taking your drug, continue on with the exact diet that you were eating before, because that was what you were sustaining your microbiome population before, if that's what you were taking in. Also things like oat bran, I think, and plain yogurt are typically seen as good food that will help replenish the good species, rather than just like transferring in good species.
Just real quickly, I will add, if you look for foods that are called prebiotics, so those are not bacteria, they're in them, whereas a probiotic is a food that has live bacteria cultured in it, like yogurt and sauerkraut. Prebiotics are foods that have structures that tend to feed good bacteria and promote the growth of those good bacteria.
We didn't get to your last part of the question too either, with like the drug specifically for single bacteria.
Targeting.
Yeah, that would be a great method, but unfortunately, the development for that is even further hindered, because then when you get to the clinics and you have someone very sick, they don't want the time to wait around to culture that bacteria and see that they have that exact specific infection. So that's the holdup with developing those types of drugs that will only target tuberculosis, for instance, or salmonella.
So I know with clinical trials for a lot of chronic diseases, there's a pretty standard way to go about planning those. Like you have your first in human dose escalation studies, you establish proof of concept, and then you move on to like the longer, more populous, like phase 2 and phase 3 clinical trials.
Mm-hmm.
So with antibiotics, because you're only taking the course for like 7 to 14 days usually, what do clinical trials look like for those?
I don't actually know if there's a lot of longitudinal studies that follow them like, 5 years after the fact.
Yeah.
But sample size is crucial there.
Okay.
You're using a lot more people for those studies.
Okay.
Yeah, so just because we're getting kind of over our time, so we probably have time for one more question right here. But then people can totally feel free to leave. But if you have more questions, we'll be lingering around here, and you can just come up and ask your questions directly. But we'll just take one more question in like this public setting, just so that people can get out of here if they need to.
What can we do to boost our own natural immunity to bacteria?
Believe it or not, exposing yourselves to bacteria. Like the very common thing is to tell your kids to eat dirt now, because that exposes them to a lot of different things in their environment. It kind of stimulates your immune system to react naturally to things. Of course, if you're immunocompromised, that's not a good thing. But if you're relatively healthy, yeah.
And, there's also your immune system, the more your immune system is taxed, the less it's able to effectively combat, say, an infection. So if you are suffering from chronic diseases, especially a lot of that are inflammatory, figuring out what the triggers for inflammation in your body are, which is an ongoing immune reaction all the time, can help you perhaps maybe if you do get a bacterial infection, instead of your immune system constantly dealing with this inflammation, you could more effectively use that immune response to combat bacteria.
Very complicated dogs then.
Very. That's why we have jobs. Trying to figure that out.
Okay, so thank you again, everyone, for coming. And if you have questions, feel free to come up and ask our two speakers who actually know what they're talking about.